Welcome to the Muscle Expert Podcast with Ben Pakulski, one of the world's top professional bodybuilders, an expert on human performance and mindset mastery. Ben dives deep to deliver the strategies of the top experts to upgrade your body, mind, muscle, strength, performance, biochemistry, and how to become the upgraded modern man. Join us on benpakulski.com to learn the cutting-edge techniques to take control of your body, your brain, and create your greatest life.
Hey, welcome to another episode of the Muscle Expert Podcast. I am your host, Ben Pakulski, and today we host Dr. Jimmy Bagley of San Fransisco State University. Dr. Bagley is the guy on the cutting edge of muscle physiology, how muscles actually work at a cellular level and how that's going to impact our training and how our training impacts our physiology. So super interesting conversation with Dr. Bagley. We kept it nice and short. We got some really great golden nuggets out of here. And some of the things you're going to take away from this episode, how training is within your control. Meaning, if you want to become muscular, your body can actually completely shift your muscle fiber composition. So despite what we used to think, Dr. Bagley is going to teach you that you actually have the ability to build any body you want, whether it be hugely muscular or conversely, the situation I'm in, going the opposite direction. It's also going to teach you the best way to implement what's called concurrent training, which means how to implement weight training or strength training with intelligent cardiovascular training, and whether it's aerobic or high intensity training you want to do. And a question I get all the time, how often do I want to do it? And how much do I want to space it away from my training? Dr. Bagley gives us some awesome insights. I know you're going to love this episode. And if you do, as always, I would appreciate a review because your reviews drive this podcast. Have an amazing day. Enjoy this episode with Dr. Bagley. What's up, ladies and gentlemen? Welcome back to another episode of the Muscle Expert Podcast. I'm your host, Ben Pakulski, and today we are hosting Dr. Jimmy Bagley, a professor at the University of San Fransisco State University, I should say. You've previously worked with Dr. Andy Galpin out of Cal State Fullerton, and you guys have a very similar line of thought. You've done a lot of similar research, and obviously you've done a ton more, which is why I was super interested because, as I said, Dr. Andy Galpin is awesome, and I know you are as well, man, so welcome.
Yeah, thanks for having me, Ben. Appreciate it.
And I'm so grateful for you guys doing this research because, as you know, man, I've been a bodybuilder for 20 years, and it's been a lot of the blind leading the blind. This has been a lot of subjective stuff, bro science, really subjective. And, you know, I'm someone who believes that success leaves clues. I believe that people who are successful in the past are successful for a reason. And, you know, there's something to be learned there, but it's so nice to have you guys out there actually quantifying all this stuff and eliminating the BS that's misguiding people. You know, there's so many BS rules in fitness that people think are true, and you're disproving them. So thank you.
Yeah, I mean, I think being a scientist, you have some level of skepticism that you're always kind of floating around with. And I think being skeptical and curious is kind of what drives me to, you know, figure out these answers.
Yeah, it's truthfully great, man. I mean, and you guys are asking the right questions too. That's what I loved about connecting with Andy was, God, you're actually someone who thinks about it. So you get so many guys who kind of get stuck in their own way, or there's this kind of, you know, pardon my language, but kind of this circle jerk of people who all just try to perpetuate each other's findings. And then somebody objectively looks from the outside and goes, man, what are these guys looking at? Like they're all full of crap. And you guys are actually looking at some stuff that's valuable and, you know, actually unbiased and not trying to be a self-fulfilling prophecy or a self-serving end result. So it's truthfully awesome to see. So the thing that's obviously Dr. Andy and I talked about was this muscle fiber typing thing. And you said you've done some other interesting stuff. You've just released a study recently about twins. Tell us about that.
Yeah. So I know most people know about fiber type that we've got fast twitch and slow twitch fibers, right? It actually is more of a continuum. So humans like to bin things, right? Like we've got this category for this category for this, get this for this. So with fiber types, originally it was binned as slow twitch, fast twitch, or red muscle and white muscle, right? Because red muscle's got more myoglobin in it, looks red. After a while, we found out that they have different biochemical properties, right? Then we kind of have a slow twitch, then we've got kind of a hybrid type fast twitch, and then a really fast twitch. And then now what we do is we use this nomenclature called myosin heavy chain identification. It's a protein. And we have type 1, type 2A, and type 2X. You may have heard 2B. That doesn't exist in humans. That's an animal thing. It's kind of miscommunication. We're actually working on a paper now to try to bring all this to light to the general public and kind of tell people where the science is at. 'Cause for the last 30 years, I feel like people have been kind of in the dark, right? And people talk about fiber types, but they're not talking about it in this long continuum of these different types. So anyway, we did this study on a pair of identical twins, middle-aged guys, healthy. None of them had any cardiovascular disease or anything like that. But one of the twins had exercised for 30 years and he was an ultra endurance athlete. The other twin was a truck driver. So total opposite jobs, total opposite, lives after the age of 20. And kind of the key take home was we found obviously that the trained twin had a higher VO2 max. So his aerobic capacity was much higher, but he also had 50% more slow twitch fibers, which was insane because you'd imagine these identical twins, they would have the same exact fiber type. They might shift 5-10% either direction, but we did not expect them to see 50% difference. So I think that just goes to show you that humans are a lot more adaptable than we always thought.
So one of the interesting things that you said that I want to pull out a little bit is you said they started differentiating at around the age of 20. Mm-hmm. So my first thought would have been maybe they started at a very young age, and because obviously we're probably a little more plastic from a very young age. Did they discuss any differences in their youth?
So we did a full, health history background. We had them fill out detailed kind of logs of what they did every decade of their life. And in their 20s, they were real similar, but they both did play sports. I think, they played soccer, basketball, baseball. It's kind of typical team sports. So and I think your point, if people exercise when they're kids, they're a lot easier to change, their body composition or fiber type or whatever as they get older. I don't have any data to support that, but I would say that if you start young and you're able to ramp up those gene process of change, I think it's going to be much easier when you're older to be able to change. Not to say that if you were sedentary as a kid, you can't adapt.
Sure.
You definitely can. My brother-in-law is in Marine Corps boot camp right now. So we're rooting for him, but he didn't do any sports up until age 18. And now he's in boot camp for the Marines and he's super fit. So, it's possible to shift too, but I think, as a kid, you want to make sure that kids are trying to adapt young.
Sure. My thought process was just going, well, maybe the one who has the 50% more slow twitch actually started more endurance at, the age of 5 or something, where the other one was being sedentary, but you said they were both active. So that kind of cancels that out.
Yeah. Yeah. So,
interesting. So, my audience is obviously interested in stuff like that. And the biggest thing that I try to teach people is the internal locus of control. And I think I said this to Andy as well, is this just substantiates that conversation where, hey, guess what? You can do this, right? So tell me what the implications are for people who want to play any sport.
So as far as if you're just starting from scratch, you mean?
Sure. Yeah. So, what would this mean for someone looking to build as much muscle as possible? Are we insinuating from these deductions that someone can literally build an unlimited amount of fast twitch muscles from what they have? So obviously they can't build an unlimited amount of muscle because the number of muscle fibers is relatively genetically predetermined. Actually, which is not the truth either, I guess, right?
Yeah. I mean, that's the idea of hyperplasia, of adding new fibers, which we're finding that probably happens in adulthood too.
Right.
Yeah, but I think people just starting out, I almost say don't overthink it with all switching fiber types and everything. I think if you start doing, so say, you're sedentary and you want to start bodybuilding, you start lifting, you start conditioning, your body is going to change. It would be nice to have like a pre-post, measure of your years long of training. But I would say don't necessarily overthink that I'm trying to work on these type 2 fibers today, because you never really know. Everybody's going to be a little different. I know my fiber type because I've been biopsied like 25 times or something. But for most people, if they're trying to train for one thing or another, I would say don't overthink the fiber type thing right away.
Okay, interesting. So if someone's looking to, just so the audience has an idea of what this actually means, can you tell us a little bit about what characteristics you see in the type 1, type 2, and type 2X?
Yeah, exactly. So type 1, these are going to be the fibers that are used, again, right now when you're sitting. Really, you got your lumbar support when you're standing, when you're walking around. And these are really oxidative, which means that they're always using fat. They're always using oxygen to produce energy. Type 2A, I think this would be the ideal fiber type for an athlete or a bodybuilder. because these are fast. They can contract fast, but they also have that aerobic or that oxidative capacity. So these are kind of like the sweet spot. And then 2X, those things are for your sprinters, your hurdlers, your jumpers. These are just, you got about 5 seconds of pure power. And those don't have much oxidative capacity at all.
Is there a difference in size between the 2 and the 2X?
That's a good question too. I get that a lot. I teach this advanced muscle mechanics class at San Fransisco State, and a lot of the animal literature shows small fibers are usually type 1, big fibers are usually type 2A, but in humans, that's not what we see. It really depends on what you do. So I typically say we're probably all cruising around the same size for slow twitch and fast twitch. But if you start training for bodybuilding, guaranteed those fast twitch fibers are going to get a little bit bigger than your slow twitch. If you start training for aerobic, some kind of aerobic event, maybe your slow twitch fibers will start getting a little bit bigger. So this is really dependent on the person. And that's what we've been, Dr. Galpin and I and a couple of our colleagues have been finding, that we can't say that, oh, you're probably 50-50. Your fiber types it really depends on what you're doing at that time. And they can shift within 4 or 5 weeks.
Would you suspect that someone with a higher percentage of fast twitch muscle fibers may be more inclined to be better suited for a diet higher in carbohydrate?
That's an interesting question.
Based on the glycolytic capacity or necessity of the fast twitch contractile fibers?
Yeah, I think that is super adaptable too. The enzymes that shift the most are actually not the glycolytic enzymes. So enzymes in glycolysis like phosphofructokinase, some of those, if you look, from trained to untrained, those might shift just a little bit, but it's actually those oxidative enzymes like citrate synthase and these enzymes involved in breaking down fats and using oxygen that ramp up a lot in your slow or your fast twitch fibers. So I don't know. I'm sure if you did have 2 different groups with different diets, carbohydrate, that it would maybe shift the glycolytic enzymes a little bit, but we don't see that too much.
Interesting. But then the flip side of that, would you suspect that someone who had a greater percentage of, I'm trying to pull something out here, like someone who had a greater percentage of fast twitch muscle fibers, how would that influence their caloric expenditure? Would there be an influence in the caloric expenditure just at rest?
I think they have more total fibers is probably what you're looking at. And actually, if you had more slow twitch fibers, so this is the weird part. So think about slow twitch. These things are always kind of on. You're using them right now. So if you have a lot more slow twitch, you're probably going to use more caloric or energy at rest, I would think.
So that's what I was trying to pull out. That was kind of the flip side of it. Is there more fat oxidation for someone who's got more slow twitch at rest?
Yeah, I would imagine. But I also would imagine if you had a marathoner and a bodybuilder, the bodybuilder may have less total percentage of slow twitch, but overall they may have more fibers or a larger size. So they're probably going to be burning more calories at rest, just the overall size.
Today's podcast is brought to you by ATP Labs. We've just released a brand new product that you guys are going to love. It's called MyoPrime, and this is just a creatine-based product with some ATP and HMB. And the reason I love this product is it's kind of an add-on to your current pre-workout. So you guys know I'm not a big stimulant person, but I am a big focus person, and creatine really helps add to that focus product that I always take. So you guys know I'm a big fan of the GF, which is the growth factor by ATP, which is primarily just alpha-GPC and tyrosine, driving up some dopamine and acetylcholine in your brain. But if you want to add the pump factor and the cellular energy to that so that you can have the fuel you need to fuel your workouts, then I highly suggest you add MyoPrime. And if you think I'm cool, I think you're cool too. So if you use the code BEN10, BEN and the number 10, 10, you get 10% off this month only. Enjoy the podcast. Yeah, this may be completely out of your realm of comfort to talk about, but how much shift have we seen in people as far as actually increasing the number of muscle fiber cells like hyperplasia? Are you familiar with anything that talks about that other than Jose Antonio stuff?
Yeah. So Dr. Antonio's got some of the stuff in bird models from the 90s, I think even, and that stuff's really cool. And now we're actually finding, so kind of to back up a little bit, everybody always thought for hypertrophy and growth, you had to have satellite cells. If these are, muscle stem cells. Mm-hmm. Now there's a whole school of thought that says maybe you don't need so many satellite cells. Maybe you can grow without it. But what we do know is that you do need those stem cells to be able to potentially split fiber split or add new muscle fibers. And there's some evidence in humans that says that we do that as we get older. Of course, we can't do like an animal, where we take a whole leg off and then we count the number of fibers. That's impossible in humans. But there's actually a study I kind of wanted to bring up from 2006 out of a group in Italy that did look at bodybuilders.
Yeah.
Bodybuilder muscle fibers. This was done by Bottinelli's group, Northern Italy. They do some amazing muscle stuff up there, and it's kind of overlooked. It's in a great article or great journal called Journal of Physiology. But what they did, they looked at a group, I think it was 5 or 6 bodybuilders. Half of them were on anabolic steroids, half of them weren't, but they kind of combined them together. And they found that compared to normal resistance trained people, the bodybuilders had 15% larger muscle fibers, but their total muscle size was 50% larger than the average person. So that, whatever 35% was unaccounted for. And they're saying that that was probably because they had more total fibers than a normal person. So were they born like that, or did their years of bodybuilding and potentially steroid use allow them to have hyperplasia or gain more cells? And I think that's probably what happened, because 35% unaccounted for size gains is huge.
No implications with glycogen retention or water retention or something like that, just from having more potential inflammation or something like that from training?
That's also a potential, I bet you, but that's maybe 5-10% could account for water retention and stuff. Because they did MRI. The best way to measure muscle size is to do a full MRI. They did slices of the thigh.
Right.
They did basically everything except for remove the muscle and count the fibers, which is impossible.
Right.
I mean, it's possible, but it's not ethical.
Right. Super interesting. This is all so fascinating to me because, as I exit bodybuilding and you start to train differently, just the idea that like you can completely polarize your life and completely shift in another direction. I think that's an empowering thought to express to the listeners that, no matter what you want to train for, it's just a matter of subjecting yourself to these new stimuli. Do you have an idea of how long it may take to actually start to see a shift? Has anyone ever started to measure, any longer duration studies?
Well, I think that the best measure for that, for shifting, or the best way to look for that quickly would be to actually take a stimulus away. And I think maybe Andy Galpin talked about this a little bit, but a lot of the studies that show the quickest fiber type shifts are from space flight. So instead of adding a stimulus, right, which is I'm walking around all day, so I'm using my legs. But if I go lift for half hour, 45 minutes, that's a small stimulus. It's going to add increments over time. So I could probably shift my muscle fibers to a faster twitch fiber type in maybe 4 to 6 weeks on Earth. But if I go to space and I deload for 2 weeks, you're going to have significant shifts, in probably less than a month. That's how long it takes for those proteins to kind of add or be taken away. So yeah, I would say anywhere 3 to 6 weeks is probably a normal time for shifting if you're really hitting the stimulus hard or taking it away.
Do we know about when in a person's life that muscle fibers start to differentiate, or does that start as early as birth?
So it's as early as birth. I mean, you're born with a lot of stem cells, a lot of satellite cells. So as I'm in my mid 30s, as you get into your 30s, 40s, and 50s, we probably only have, if all of our nuclei, say we have 100 nuclei in one area, probably 2 or 3 of those, maybe 2 to 3% would actually be stem cells that can be used to repair and add new nuclei. But little kids, I don't really know the number in humans, but it's upward of 20+%, I would imagine, of actual stem cells that are working. So they're super adaptable, super plastic when you're really small, and those fibers are differentiating at least until you mature. But then I think still there's ability to have hyperplasia. We just haven't had that definitive proof yet to show it.
So can you walk me down do we know anything about the mechanism of how these things actually shift? Is it just a matter of like changing the hemoglobin structures in the type 1 to type 2? So yeah,
so when you talk about shift, what we're actually talking about is that protein shifting from a type 1 to type 2A protein. So it's the same protein, right? But it's just a little bit of conformation change. And to do that, there's a lot of different kind of a cascade of events that nobody actually knows for sure. But what probably causes the shift is we've seen, so there's also these hybrid fiber types, which is a whole nother can of worms. But you have some fibers that are type 1 and type 2A on the same fiber. But what we've seen is, so each muscle fiber is innervated by a motor neuron, right? So your brain tells your bicep to curl. You're doing that all the stimulus is happening there, but each fiber only has one innervation. What happens is if you keep stimulating that to lift heavier and harder, maybe those type 1 motor neurons start lifting off of that site, and the type 2A neurons drop down and start innervating that fiber, and then it starts to act more like a 2A. So kind of what we know is that it's a top-down thing. Like, I'm a muscle guy, so I don't know anything about the brain. Don't ask me any questions about neurophysiology or anything, but I do know that it's probably your brain telling your muscle what to do, and those fiber types will shift depending on which motor neurons attached at that time, and that can change. Yeah.
So if someone's trying for optimal hypertrophy, which I guess a lot of my listeners would be, would it be suggested that they would want to stay? Like what type of rep range, what type of time range would you suggest if they're looking for getting the greatest amount of shift toward type 2 fibers, type 2 and type 2X?
Yeah, I think with the rep range stuff, there's been some cool stuff. You've had Brad Schoenfeld on your podcast before, and he's the rep range guy. So I would ask him more specific training questions about that. But I do know that if you want those 2A fibers, the kind of the goal fast twitch fibers, probably your typical bodybuilding, maybe like 8 to 12 reps, multiple 3 to 5 sets, like your standard bodybuilding.
Is there an amount of time? Like, can you walk me down the physiological process, the physiological response? So let's say I'm doing a bicep curl, and it's relatively light, it's relatively low as far as my perceived exertion. What's happening inside a muscle?
So if it's relatively light and low, like what we know is that there's 3 motor neuron types, right? There's kind of go small to large, depending on how big the neuron is, the axon of it. You're probably only using those small neurons at that time. And those are connected to your slow twitch fibers, maybe a couple of those fast twitch. You start increasing the load. You have to add more motor neurons. So you're not even able to activate those 2X fibers until you're working almost maximally.
Now, do you think it's a load thing, or is it a fatigue thing as well?
I think the fatigue. So also what happens too is it's not like you're just firing it continuously. They're kind of shifting. So maybe one slow twitch is firing, the next slow twitch is firing. As you get fatigued, I think that firing rate starts to get a little bit messed up. And I still don't think you'll be able to activate those 2X fibers, even if you're super fatigued. You still got to add that stimulus with that extra load. So this is a whole nother idea of there's 2 ways to overload. There's a metabolic overload, right? And then your mechanical overload. I think to hit those 2X fibers, the metabolic overload's not going to cut it. So like your BFR training, stuff like that. If you want to activate the 2X fibers, you really have to go fast and or heavy.
Is there anybody studying that specifically? So there's a good friend of mine, Jacques Taylor, who's been on my podcast as well, who's doing a lot of work in this stuff. Trying to determine, or has determined from his perspective, that you can actually recruit type 2s with fatigue as well. But I'm curious to know if you know of any studies that suggest either way.
Well, I'm sure you can. Like I said, you can probably recruit those 2A fibers, but most researchers don't even differentiate between the 2A and 2X fibers because they're so close that they functionally are so different. The only study that I've actually seen that had an athlete with a lot of 2X fibers was a study that came out recently. It was a sprinter or a hurdler. He was an Olympic hurdler, and he had upwards of 30% 2X fibers. That's probably genetic, right? But that guy doesn't do any metabolic. That guy hasn't run a lap around the track his whole life. He pretty much just does 100 and he's done. So I think for those 2Xs, those really super fast twitch fibers, you really got to do something that's a mechanical overload, or just be genetically, gifted having them.
Super interesting, man. Yeah, that stuff's all very fascinating. And now another thing you brought up is integrating these things with some type of concurrent training. I know it's another area of interest for you. Tell me a little bit about that.
Yeah, so I grew up trying every sport possible. I was horrible at most things. I think everybody kind of finds what they're good at. And I ended up being good at swimming and water polo. So I'm a water guy. But doing that, right, we're in the water all the time. We're pretty much overtrained all the time. And I felt overtrained up until I was like 22 years old. Then I got out of the pool and I was like, dang, I can actually put some mass on, right? Like I was a skinny twig until I was 22, even though I was lifting 5 days a week. I was just totally overtrained because, we're in the pool, at the gym, in the pool, back to back. So at age 22, I started thinking about it and like, I still want to do aerobic exercise, but I also want to get some gains. I want to get big arms. I want to look good too. So I kind of started thinking about this concurrent training and how we can best do this. And a couple of years ago, my friend Kevin Murak and I, Dr. Kevin Murak, he's at University of Kentucky, wrote a paper, and it's called Muscle Hypertrophy with Concurrent Exercise Training. And we basically looked through the literature to try to find out the best way to maintain your gains or get gains and still be able to aerobically train. Because, we want heart health. I want to be able to climb a flight of stairs without getting gassed. I want to be healthy. So we scoured the literature and we actually found that there's this kind of misnomer. A lot of people think that if you do both of these, you're not going to get gains. If you do aerobic training, running, swimming, biking, whatever, and lift, you're just going to be blunted. But it really depends on a few things. It depends on what type of training you're doing and the timing. The timing is really important. We looked through a bunch of studies and found that people were able to get the most hypertrophy along with their concurrent training if they spread out those training sessions by at least 6 hours up to 24 hours. So that's what I do. Right now I'm not training for anything super specific, but I'm lifting Monday, Wednesday, Friday. Tuesday, Thursday is my swimming or my running days. And then Saturday, I'm either surfing or doing a track workout or something like that. So, and I feel way better than I did when I was training 3 times a day.
Yeah. Can you tell me why that is? Any idea?
Yeah, the idea is, so there's this thing called the interference effect. So you've got kind of gene programs that are geared towards size, strength, and gene programs that are geared towards endurance. And the idea is if you overload the body with a bunch of these endurance genes, that it'll kind of override your strength or size gains. And I think that's true to an extent, but by spreading the time out and giving your body a chance to recover, I've been involved with several. We're actually doing it right now with Dr. Galpin, some time course studies where we look at these genes being activated after exercise. What's the time course of them ramping up and then coming back to baseline?
Beautiful.
Right. Most genes go back to baseline within 24 hours. Some of them will stay elevated, but the ones that we really care about, 24 hours, you're almost like reset and you're ready to go for your resistance training. You do that 24 hours later, you're ready to go for your aerobic training.
So then when you say 6 hours, that's definitely the low end. You ideally want to be closer to 24 if you're looking for maximum hypertrophy. Right.
So it's almost like between 6 and 24 hours, there wasn't much difference between sizes. But when you get to 5 hours difference, 4 hours, 30 minutes, like I used to do, go downstairs, lift, go up to the pool, swim. That's probably not what you want to do if you're trying to get size. So for me, it was all I had, as a professor, I've got my class schedule and everything. So I only have a certain amount of time to exercise, but I would say at least spread it out. And then again, you've had a lot of people on your podcast talk about nutrition timing, have the meal in between or, right around your first workout.
What's the most exciting thing you're teaching right now as a professor?
Oh man, I get to teach the coolest stuff. I get to teach exercise physiology and muscle mechanics. And basically I get kind of the freshmen, sophomore, juniors, to get them really excited with XPhys. And then by the time they're seniors or grad students, I get to take them through this class called Muscle Mechanics, which is an advanced, basically all we talk about is everything from the molecular cell level, like we're talking about here, all the way up to the whole body and how it all connects. And that class I've been teaching for 3 years, and every semester it keeps getting better and better and more fun.
So give me the 3-minute or the 5-minute synopsis of what you're walking through in that class.
Okay, so we've got a few different kind of motifs in the class. The first one is that structure equals function. You probably heard this if anybody's taken a biology class.
I say it every day.
Right? So your muscle fibers are these long cylinders. They contract. If they were round balls, you wouldn't be able to, move. So structure equals function all the way down to the gene level, all the way up to, you can tell the difference between an elite marathoner and an elite sprinter, structure and function. So that's one thing we always go through. The other thing is kind of this small to big or big to small. We look at everything from the gene level. So we're studying these proteins called AMPK as the energy sensor of the cell. That's, involved in your aerobic energy pathways. How that affects everything all the way up to your marathon time, for example. So we go through all those and then.
So again, that's interesting to me, man. So. And Andy and I talked about that a little bit, and we talked about MTOR and AMPK. How is that, you said, influencing your marathon time? Obviously, that's a bit of a stretch. It may take a while for you to walk down that road, but walk us down a little bit give us a little bit more insight into this AMPK thing. Cause like I said, Andy and I kind of breezed over it, but I'd love to get your insights because it's super interesting. Like the MTOR AMPK balance is something that is this massively misunderstood or just completely not understood thing in our world. So we're all concerned with, I want to go in the gym, I want to build muscle. Well, unless you're, from what I know, unless you're stimulating mTOR, you're not building muscle. So, and conversely, if you're trying to lose fat or if you're trying to maybe break down tissue or improve your endurance capacity, there's got to be an AMPK stimulus. Can you teach us a little bit about that?
Yeah. So, kind of like we talked about, there's these separate gene programs that you mentioned. Without going down the cascade, that mTOR and AMPK, that's where we get that idea of that interference effect. That AMPK blunts your mTOR response, and that's why we separate things. That's why you eat, because eating is going to stimulate the mTOR response a little bit. The AMPK is interesting. So we call it like the energy sensor of the cell, the gas gauge of the cell. This also, it's in every cell in your body. It's in every cell in I'm pretty sure every eukaryotic cell. So every animal has AMPK in there, and it's really important. So when people think about it, they're like, "Oh man, I'm stimulating AMPK. It's going to mess up my gains." Not necessarily. Your AMPK, actually, getting even deeper, it has multiple subunits on it, which we're studying. One of them is a catalytic subunit, so the one that gets activated. That's alpha subunit. There's the beta subunits, the scaffolding, which is kind of its structure. We're not super interested in that. And then the gamma subunit's the regulatory subunit. And we're finding that these are different between athletes and non-athletes, between pre-diabetic, diabetic people and athletes, between fast twitch and slow twitch fibers. So it's not just whether you have AMPK ramped up or ramped down. It's which part of AMPK, how's it working to interact with everything else. It's just, man, it's pretty crazy to think about all this stuff. We just keep opening doors and we're like, now what? And we just open the next door, and then now what? Now epigenetics, now this, that. It's like, we're never going to be out of a job. The scientists are always going to have a job at least.
So where do you see that going? So like, have you played it forward 5 and 10 years for your career? And where do you see your greatest area of interest?
Oh man. So right now, like, I'm in my third year as a faculty at a university. I'm a yes man. So I'm saying yes to everything. I'm exhausted. I've got like 14 projects going on. I'm excited about all of them. But I think as I get more honed down to what I want to do, it's kind of related to this concurrent training stuff and looking more deeper into AMPK, kind of getting into, I don't know if you've talked about bioinformatics at all, but instead of just looking at one targeted gene, now there's pieces of equipment you can buy, set on your desk about the size of a laptop, that can give you the gene expression for 500 genes in a few hours. And then taking that information and then seeing how that compares to, again, performance. Or I'm also really interested in looking at the cell using a microscope called a confocal microscope, seeing how that affects the structure of the cell. So I think for me, it's really tying it all together, using the expertise that like Dr. Galpin has, looking at these proteins, and my expertise, looking at them visually with a microscope, kind of tying it all together and seeing how it affects people's performance. Yeah.
So when people are training, how much consideration, so you kind of, like I said, breezed over this, but how much consideration should they be placing on the mTOR and AMPK? So can we influence it? And obviously we can, but can we look for some phenotypic expressions or some physiological expressions in our body that may be indicative of one or the other?
I think one thing is that, everybody always hammers home is listen to your body. If you're tired, a couple weeks ago, I had done 3 days back-to-back workouts and I was exhausted, and I was like, man, the AMPK must be ramped up. I know you kind of, like, as a scientist, you're just kind of thinking about what's happening. Sure. But I think if you're tired, your muscles are cramped, you're sore, there's a lot going on, but that might be like, you need to work on taking the volume down a little bit. So I would say, listen to your body, see how you feel. You're not going to know exactly what's happening with AMPK in your own body, but if you're tired a lot, then that means you maybe need to back off the volume a little bit.
So what are the main factors that are influencing, let's start with the AMPK. Is it water? Is it hydration? Is it electrolytes? Is it just glycogen and ATP? Do we know what the signaling pathways are there?
Yeah, I think it's really just the immediate energy availability in the cell. So if you have, ATP is the energy currency of the cell, that drops. As soon as that starts dropping, AMPK is like, whoa, we got to start conserving energy somehow. So it's pretty immediate, really. Like you'd have to do, that's another limitation with our studies is we're doing muscle biopsies. We're not looking real time in your quad as you're lifting to see what's happening with AMPK. We only have certain time points. So we might get a pre-exercise time point, immediately post-exercise,
Sure.
30 minutes, 4 hours, whatever, but however many biopsies you want to go through.
Yeah. Andy and I were joking about that, man. I'm definitely not a volunteer. You've done 25 of them?
Yeah, I lost count after a while because sometimes, you have the same incision and you'll use that incision multiple times. It's you don't feel anything. It's like lidocaine. So you get a little bit of basically xylocaine, lidocaine on there to numb it up. But yeah, 25 plus biopsies. Again, it's real small, about the size of a pencil eraser, like less than 100 milligrams of tissue. So you're not going to miss it unless you're super elite athlete. Maybe you might miss that little amount. But
is anybody doing research into the implications of ketogenic dieting on its influence on AMPK? So obviously with ketogenic dieting, I would presume our glycogen stores are going to be relatively low. Our ATP stores are going to be relatively low. Is that going to be influencing the cell's ability to stimulate mTOR, from your opinion?
I haven't done the research on this specifically, but I'll bet you, I mess around with my diet all the time too. So I've gone a couple weeks at a time, totally ketogenic. You notice when you first go keto, right? That first couple days, you're okay. Maybe day 3, 4, and 5, you start feeling really crappy. You get that acetone breath, the horrible bad breath, and it kind of goes away. I think when you're going through that point, when your body's trying to ramp up producing its own glucose and everything, that's probably the time when AMPK is freaking out a little bit. But I think, you can adapt to anything. You've got, Native Americans in Alaska that have lived off of just blubber for, years. So people can live off almost anything. Your body will adapt to it.
But they're also not athletes trying to hypertrophy their muscles.
True. That's the thing too. So I was trying to go keto a couple weeks ago and I had a 5K, whatever, it was like a Sunday. I was just running for fun. And the day before I was like, I got to have some carbohydrates, man. I'm like, it really depends on what you're trying to do. If you're trying for performance, your diet's going to have to get really dialed and you're going to have to be really used to it. But for a normal guy like me, I'm just going to kind of play with stuff and see how it goes.
Yeah, I'm very curious. And if you hear of anybody doing research, I'd love to hear, how long it's taking for people to adapt to produce their own glycogen in these ketogenic states. Because obviously keto is massive right now and everybody's jumping on the keto bandwagon. There's a lot of misunderstanding, I think, or lack of understanding around performance. Like, I don't think we can refute that it's great for your brain. It's great for your body. It's probably great for longevity. But performance is what we're after ultimately, or what I'm after, my demographic is after. And it'd be awesome to know if you can actually sustain that high level of performance. Any opinion and speculation on that?
Yeah, I think, again, probably some people can. I know I can't, though. Like I said, I had to have some carbs the day before my 5K or I would have been crushed. I know I would have been crushed. I think some people can. And I think you're probably not going to find that many Olympic athletes that are going keto during their competition. Right. Like I think throughout the year, you might phase in some of this stuff, but I don't think it's going to be something where most people are going to be doing around a competition.
Fascinating. So walking down the road now of MTOR, can you tell our audience a little bit about what they need to know about MTOR and how to influence it?
Yeah. As far as the cascade and everything goes, there's plenty of other scientists that can talk a lot more about that than me. But I would say, again, with MTOR, it's all about like that timing. And again, your peri-workout meal around your workout.
So is it? I want the energy status to be high of the cell. Like, what are the main kind of catalysts for that?
Yeah, you want the energy status to be high. You want the body to think that, okay, I'm ready to pack this stuff away. So that's why when you're tired, I'm saying like mTOR is going to be inhibited if you're feeling tired, because that's going to mean that your body's not ready to pack anything away. Without, again, getting too into all this, people think, this was in my dissertation. I remember one of my advisors was like, what's the most anabolic hormone in the body? And I was like, oh man, testosterone, obviously. He's like, nope. I was like, growth hormone? He's like, nope. I was like, anabolic. I was like, insulin? He's like, bingo. So the most anabolic hormone in your body is insulin. Its whole job is to pack everything away, store it away. This is where, think thinking with not really performance related, but more like health related, stimulating AMPK, you need to do that or else you're going to become a diabetic pretty much. So don't like push that to the side. You're going to need to do something like that too. But if you're really focused on mTOR and getting that going, just make sure that you're fueled correctly. And again, that's different, a little different for everybody. It's going to be a meal around your workout, and it's going to be spreading out your workouts.
I know you've briefly mentioned some other scientists doing some interesting work. Who is your greatest area of interest right now? What scientists are doing research that you're really into?
So, I mean, there's always these kind of hot new topics going on. And I've always been, like we talked about a little bit ago, I've always been really fascinated with hyperplasia, muscle fiber splitting. And I'm like, we need to find the answer. I want to see that in humans. Also along the lines of how that is influenced by satellite cells, these muscle stem cells. Well, there's, again, these two kind of schools of thought. One in Scandinavia, I believe it's University of Oslo, Brusgaard is the lab name. And they are basically saying that you need satellite cells to be able to have sustained hypertrophy. You need stem cells there. And then there's this other group in the University of Kentucky and some other places here. I mentioned my friend, Dr. Kevin Murak, and they're saying maybe you don't necessarily need these stem cells for sustained hypertrophy. And this is kind of like this, almost nerd battle, right? They both have their models and animals and human models, and they both have valid points. And I just enjoy going back and forth reading their articles come out, and there's always rebuttals. Have you seen those letters to editors and journals, and it's like a rebuttal, and it's like, well, they had made this point, but we make this point. And I think science is fun when it gets heated like this, when there's these debates that are like, we don't know the answer, but these people are putting millions of dollars and they have some of the smartest minds out there trying to figure out, these basic questions that will ultimately help athletes and help normal people. It's going to take a while to get there, but yeah, this is the cutting edge.
Are we actually doing human studies on hyperplasia?
No, man, that's it. They're all animals and cells because they can manipulate them the way they want. With humans, we're getting better at doing some imaging techniques where we can maybe count fibers. Like if you do an MRI on the leg, you can't count muscle fibers. We've got to get to a better way where we can actually count the fibers or get a biopsy right at the right time point where we see these fibers splitting in humans. I think we're getting there, but technology's got to advance a little bit more.
So is that just a matter of like someone volunteering to go to the lab and say, hey, you got to be able to prick my leg or whatever right after my workout and see if these satellite cells are actually there?
Well, we know that satellite cells are activated after workout. We know that satellite cells are related to hypertrophy. We just don't know if they're needed for hypertrophy, right? And that kind of is good for me and for anybody in middle age because your satellite cells start to drop off. Unless you do something like anabolic steroids, that's going to bring it back up. Again, that's one mechanism to grow. But these groups in Kentucky and other places are saying maybe we don't need that to grow. Maybe there's other factors inside the muscle fiber itself that can allow the fiber to sustain growth. So, I mean, it's still like a little foggy, but I'm enjoying watching that battle go back and forth.
You said you've got 14 different studies going on. I don't know if you're being
no, it's true.
Facetious. Yeah. So give me some ideas of what you're working on.
So I kind of bin my research. Again, humans like to bin things like fiber types. I've got methodological advancements. So methods that we're developing, whether it's, determining how to look at fiber type better or whatever. So I've got these methods papers, and I've got these applied sports science stuff, which, I mean, you guys would be really stoked on probably looking at blood flow restriction training and bicep curls. I've got a student doing that. I'm like, this is a sweet study. Let's do it. I've got a student looking at compression socks, students looking at stuff like that. So this is kind of my applied area. And then also this basic muscle physiology, looking at fiber types in different athletes. We're actually even comparing fiber types from people that have been on dialysis and kidney failure for a decade. And man, their fibers are messed up. They look bad. Comparing that to a normal person, comparing that to an elite power lifter that's been taking anabolic steroids for 4 years. I mean, we've got the full gamut. We're one of the few labs, again, us and Dr. Galpin in the world that have access to this tissue. So
what's the limitation in science right now holding us back from really, putting all these pieces of the puzzle together?
Man, so it's not ideas. We have a lot of ideas. I think it's really time and money. I mean, I mentioned I teach too. So I teach 3 classes a semester. That's in itself a full-time job, plus trying to hammer out all these studies. I think time and money, and I think collaboration is really key. So the way that I work with Andy's lab, we also work with some folks at Cal Poly Pomona. Dr. Eddie Joe's out there. He's awesome.
Yeah, he's awesome.
Yeah, we work with Dr. Josh Cotter and Evan Schick at Long Beach. So we've got kind of this group in California where we call ourselves like the Muscle Research Consortium, right? We share data, we share muscle tissue, we do all this, but we also share the rewards, right? So we share the publications. Everybody's got ego, right? But we try to put our ego aside and bring the team together. And it's way easier said than done, even though we get along great. Everybody's got their own stuff going. And, it's hard just to get a phone call with 5 people together, right? Let alone write a 40-page paper. So,
right.
Yeah.
That was the objective behind my podcast when I started, man. It was nothing to do with me. I never claimed to be the muscle expert. The idea was to get people like yourself on and give you guys a platform to collaborate. And then, hopefully somebody else hears and goes, oh, I'm working on that too. Let's figure out a way to add to that. And then we have this cumulative effect. And it sounds like you guys are doing that already. Because ultimately, man, this is where we got to go, right? I really believe in it, and this. Like we're so primitive with training methodologies, with our understanding of these things. The fact that 90% of the fitness community just doesn't understand, and the common beliefs and common understanding are usually misconceptions. So I'm very grateful for all the work you're doing, man, and please keep doing what you're doing. Can you give our listeners a couple final tips on how you might recommend manipulating their training to build their greatest body, whether it be, let's say muscle building? If someone wants to be big and lean, what tips would you have for them to lean toward that direction?
Man, it's going to be the same stuff that everybody kind of always says, like work hard, all this. But I always say, when I tell people, work hard, but also stay humble. And I think bodybuilding is a sport that will keep you humble, right? You're always going to come up against a wall. You're always going to come up against something. Same thing when I was in swimming and water polo and stuff. You got to work hard, but also stay humble and take your failures take them or basically look at your failures, or look at times when you're feeling down and stuff, and use that to build yourself back up. So an example I use in my own work, I get rejected a lot in research. It kind of sucks. I mean, it's going to really suck when you work really hard on this paper, or work really hard on this training program, and it just falls flat. You don't have the body comp you want. Or for me, I get a paper rejected here. Pick yourself up, go try again. If it's not working for you, if you're training, you're not down, you want to be lean, whatever, 8% body fat, go back to the drawing board, keep testing, keep working. Yeah.
All right, man. Nerdy stuff aside, what are you thinking about when you're doing a 5K?
Man, I'm kind of a Zen runner, so I don't run with any watches or anything. I sometimes wear a heart rate monitor, but I am thinking about basically my breathing most of the time. And I've always put a song up in my head, man. It's usually something a little more upbeat. Sometimes I like surfing and all that, surf rock, Jack Johnson. I'll just play a song in my head over and over again. Yeah, I like running. 5K is about my distance now, though. I'm not going to do any more marathons. I'm done with all that.
What's the hardest thing you've ever done?
Hardest thing I've ever done physically?
Sure, or mentally.
Yeah, mentally, freaking my dissertation for my PhD was pretty tough mentally. But physically, I used to be a beach lifeguard in the Central Coast in California. And man, it was called our Hell Week. So every season before we were all out of shape, we would go out. There was one day and it was the waves, I think. I always over exaggerate, but probably double overhead, 12, 14-foot waves. We had to do a swim, run at the beach. I thought I was going to drown. I was crushed. It was like, a mile run, swim out a quarter mile back, and do another mile run. On the coming in, I thought I was going to die. That was probably the hardest thing I've ever done.
Nice, man. Jimmy, I appreciate you, man. Thank you so much. And I look forward to having you back on. So like I said, with this muscle expert thing, nothing to do with me. I'd like to get people like yourself back on every few months. Just give me updates on, hey man, here's what we're doing right now. Here's what I'm excited about. And another thing that I want to offer you is an opportunity to give a shout out to our listeners. If there's anything you need at your lab, if you need research, or if you need students, if you need research assistants, if you need people to be in studies, if you need donations or whatever it is. Like if you have anything that you need at your lab, this is why I ask, like, what's holding you back from really pushing the level of knowledge up? Because ultimately that's what it's about, right? Let's create a collaborative where people can pitch in some way if they happen to be out in San Francisco, or if they want to come down to your area and contribute as anyone or anything you need.
Yeah, absolutely. I mean, feel free. If you're in the San Francisco Bay Area, come out to San Francisco State. We're right in the city, about a mile from the beach. Come and check out our labs. If you go to my website for my lab, it's musclephizlab.com. At the bottom, there's a little button that says donate. You can click on there. If you'd like to donate any money, that goes directly to our lab. We use it to help fund master's thesis. So, right now I've got 4 or 5 master's students coming through, and they need disposables. They need gloves. They need, random stuff like this, or bigger items. But just helping out that way would be great. And then check us out on social media too. So it's at Dr. Jimmy Bagley, and then at Muscle Phys Lab on Instagram and Twitter. And again, we're happy to give tours. We are looking for master's students and even research techs. If you have any interest in muscle physiology and just want to come check out the lab, come on down.
Awesome, man. Thank you so much, Dr. Bagley. I appreciate you, man.
Cool. Thank you.
So what'd you think? Dr. Bagley is obviously a super bright guy who's right in the trenches there in San Francisco, and he calls you out. Come out to San Francisco and visit. You've also got his friend, Dr. Andy Galpin, down in Cal State Fullerton. These guys are doing some amazing stuff that are literally helping to drive our understanding of how to do this muscle building thing, and most importantly, how to make it easier, make sure we make less mistakes. And that's what I'm here to do for you guys. I never claim to be a muscle expert, but what I do claim to have is an amazing network of people that give you guys incredible insights and intelligent research to how to make your life great. And if you appreciate that, I would appreciate a review. I would appreciate a share with anybody you know that's going to love this muscle building journey and the vision of making you live your greatest life in your greatest body. I appreciate you all. Have an amazing day.